Hi, this is Beverly, recording the genomics lecture for week 13, and it's on genomics. So we know that genomics and genetics are words that you often hear and people use interchangeably. Genetics is a new science, hasn't been around for very long, but just so you know, the science of genomics is even newer. The genome was coined in the 1920s, and it was referred to as a complete set of chromosomes, and currently it refers to the DNA and haploid sets of those chromosomes. The science of genomics was started in 1986, and it is the study of genomes. Okay, so how do we study that? We develop linkages, so we use genetic maps, and those can be cytogenic maps, linkage maps, physical maps, or sequence maps. This here is a pictorial accompaniment of those particular types of maps that were just said and how more specific they become as you move down the list. So we need to talk about the term positional cloning. Positional cloning is where things are looked at by a gene approach, and we look for matches in those single genes, and we look at how those relate to specific diseases. It starts with a phenotype, an expression, what is the causative agent or the gene, and then we locate where that gene is on a particular chromosome. In the 1980s, this helped us discover diseases such as Doshman's muscular dystrophy, cystic fibrosis, and Huntington's disease. Um, so we know about the Human Genome Project, which started in the 1980s, and it sequenced the human genome. There was an officially started in the 90s, and it is a 15-year project. The first draft of the human genome was in 2001, and the finished sequencing of the human genome was in 2003. There are lots of workers and researchers that went into this process, and it is consists of an international collaboration. It looks at recognizing the impact of the human genome on public policy. We need to take a look at how that affects our policy. About 3% of the budget was used to address the legal, ethical, and social issues related to this. And this is to help prevent the misuse of this genetic information and genotypic discrimination. So there have been some interventions. So there's express sequencing tag, which is a type of technology that helps researchers locate protein encoding genes, and it looks at these coding genes and how they are expressed in a specific or particular cell type. We've also seen some key inventions of the DNA microarrays, which we talked about earlier in the semester. This displays short sequences of DNA molecules, and it helps with sequencing and gene expression. We also know that the project helped researchers cut the genome's worth of DNA, and they looked at overlapping. We know that we can randomly cut small fragments and they can be sequenced. We know that we can use computerized algorithms to search for these overlaps. And looking at the overlaps and using software, we can look at the entire DNA sequence. Our government helps fund an international consortium. We look at aligning these pieces at one chromosome at a time. And there's a private company called GENOMICS, which has done the whole genome, where they shattered the genome and then they rebuilt it piece by piece. So what is deriving a DNA sequence? This is what it looks like. We look at random fragments first. We align them up to look for where they repeat, and then out of those repeats we develop the sequence. So this is a pictorial of the mapping. This is another pictorial that looks at a more focused step-by-step direction to it. So we're going to sequence it, so we're going to break it all apart, and then we have to assemble it. So we're going to put it through this automated DNA assembler. Then we're going to look at the annotation, so it looks for clues of where the protein encoding genes are, and then we're going to put these into those tiling microarrays. for gene expression. So sequencing methods were first developed in 1977. He termed it chain termination method, and this was by using chemically altered bases to look at DNA synthesis fragments at specific base issues. New ways that we look at it is that size separated and DNA sequencing can be read, so we have better and more efficient techniques than what was developed in 1977. This is another way that we can get the DNA sequencing data. This is by the different 4 proteins, each represent a different color along the segment of DNA that's right there encoded to the protein level. This is a way that we can read DNA sequences within the science of genomics. So they are ordered by size, and then the bases are highlighted, and then those bases are pulled to create a sequence. So as you can see, there's all the T's, all the A's, so where they repeat. So this is now our number 3 is our sequence. This is just a picture of a DNA. So sequencing methods, so some of the new approaches use microfluidics. This method is called the 554 sequencing, and it can sequence up to 20 million bases in 4.5 hours. There's also a new approach called nanomaterials, which is able to distinguish how each of the 4 bases disrupted within an electrical field. This picture here, just want you to be familiar with some of the information on here, and it's also a good idea, just kind of historically to see where we've come in terms of genomics and the Genome Project. So comparative genomics, okay, so 100 species have had their genomes sequenced. Some of the first were bacteria and viruses. The very first to be completely sequenced was influenza in 1985. And then we've looked at our closest relatives, such as rats, chimpanzees, and scientists have looked at some of our favorite things, such as cats and dogs. So some of the most informative information we've got helps us represent the evolutionary crossroads. So where there are intersections within different species within the DNA genome sequencing. We can also look at modern species. This allows scientists to infer perhaps evolutionary relationships from DNA sequences that are conserved and presumably selected over time. We look at some ways of looking at these sequence similarities in this next pictogram. So if we look at some sampling of animal genomes that have been done, they've done the wallaby, the fowl, a dog. So what might it answer for us if we look at this comparative genomics? So looking at, say, a dog and a wallaby, or a human and a chimp. So they're looking at what is the minimal set of genes we need for life. They're going to look at the distinction between the 3 domains of life. So what is the simplest urocate that's found? Is there a basic blueprint for all animals? What are the genes used for terrestrial living? How do we look at birds to mammals, chimps to humans? It just is helping science put together some of these questions that they've been asking themselves. So we know that there's a human exome, which includes DNA sequences that are transcribed into RNA and then translated into protein. There are 180,000 exons. So we look at single repeats. We look at ideals. We look for insertions and deletions. And they also analyze and confirm extended observations. Could tell us very little about our own genome-encoded proteins. Researchers are studying subsets of these, such as lipidomes or secretinomes. Other nomes have to deal with transcription, disease, and pharmacogenetics. And there is a global effort worldwide, which is called the Human Variome Project, so that researchers can put their data into an international database, which may assist other researchers and inform other researchers of the work that's been done, and the questions or answers it has given us. So there's also the human microbiome, which is our body has other omes other than the human genome. 90% of our genomes are from microorganisms. And there's a human microbiome project, which is looking at 5 body parts to analyze the complex difference among individuals. Perhaps why does one person develop MRSA from an exposure and maybe another person does? Is there something within their own that prevents this from happening? This here is a patient pedigree. We've not really talked about these in throughout the thing, but when we look at family histories, when we look at a pretty serious one, we look at whether or not people have certain diseases. So we know squares are male, circles are female. Um, the line through it generally means that perhaps they've passed away, that they have passed away. And this one is done with that it has different diseases, represent different colored little squares in the or triangles within the circles or squares. So it's looking at what people had, what diseases, where did they go generationally, and how that might all be interconnected. This is another pictorial that looks at our gene and environment interaction. So how do certain environmental things impact different types of diseases? So maybe we can modify some of these environmental things and lessen the impact. So there is personal genome sequencing, which is this last slide here. Um, and I'm going to pop it back up. I'm sorry, I lost the last slide here. So we're going to put this last slide back up, and it talks about personal genome sequencing, which is really comparing genomes of healthy people with certain diseases. And the goal is to look at how those genes affect health. And it might help us to assist in recognizing the controllable environmental factors and then be able to improve someone's health. Currently in research are sequencing of personal genomes so that the information can be used in several ways, but that's not complete at this time. Thank you.